Biological control is a pest management method that uses natural enemies as control agents. Biological control operates through naturally occurring trophic interactions, which is an ecological term that refers to the general position of an organism within a food web. There are 4 basic trophic levels. On the first level, there are the autotrophs, organisms that can create their own food, like green plants that produce energy from the sun. On the second level are the first-order consumers, herbivores that obtain energy by feeding on the autotrophic plants. On the third level are the second-order consumers, carnivores that obtain energy by feeding on herbivores. At the fourth level are the third-order consumers, which are top predators that feed on other carnivores. Not all organisms fit neatly into these categories, however, as many species are omnivores that feed on both plant and animal material. The interactions between and among organisms at different trophic levels are natural aspects of a healthy ecosystem that keep conditions at a stable equilibrium. Populations of herbivorous insects at the second trophic level are usually kept in check by natural enemies at the third trophic level. These natural enemies can include predators, parasitoids, parasites, and pathogens such as bacteria, viruses, and fungi. If the pest is a weedy plant species, even the herbivores feeding at the second trophic level can be considered natural enemies in biological control systems. Biological control aims to exploit the activity of natural enemies in order to manage a target pest or complex of pest species. The most common way to achieve this is to introduce, augment, or conserve populations of natural enemies within a managed habitat to promote their activity in pest suppression. Natural enemies that are recruited by humans to reduce populations of pests are referred to as biological control agents. These agents are recruited to attack and kill pests. In the context of this course, we will consider 4 types of biological control agents. The first 2 are parasitoids and predators, carnivorous organisms, many of which are arthropods that feed on and kill arthropod pest species. The third group consists of pathogenic microorganisms, such as bacteria, fungi, and viruses, which are important biological control agents against a variety of pest species. The final group includes herbivorous insects, used as biological control agents in the management of invasive weeds. Let's begin by looking at parasitoids, the most widely used biological control agents. Parasitoids are insects whose larval stage parasitizes and eventually kills its arthropod host. This is in contrast to parasites, which do not normally kill their hosts to complete their life cycle. Interestingly enough, all parasitoids have free-living adult stages that do not feed on the larval host. Most parasitoids lay eggs on or inside the bodies of their arthropod hosts. Upon hatching, larval parasitoids feed on the internal organs and hemolymph of the host, eventually killing it. After juvenile development is complete, the free-living adult parasitoids emerge to mate and seek out a new host. Many parasitoid insects belong to the orders Hymenoptera and Diptera. Parasitoids can be specialists that target specific host species, or generalists that exploit multiple hosts. The host-finding ability of parasitoids makes them effective control agents in IPM systems in which managers try to minimize impacts on non-target species. Only the ovipositing females will contribute to pest control, and their reproductive capacity is dependent on host availability. It is estimated that over 65,000 species of Hymenoptera and 15,000 species of Diptera are parasitoids. Many biological control agents used all over the world are from these orders. There is even one insect order that is comprised only of parasitoid species, called the Strepsiptera, or twisted wing parasites. This small order contains approximately 600 species that parasitize insects in 7 different orders. The best-known strepsipterans are parasitoids of insects in the orders Hymenoptera and Hemiptera. The use of strepsipterans as biological control agents is extremely rare compared to other parasitoid groups, although their biology is rather notable. In most strepsipterans, the free-living males seek out and mate with pathogenic females, which never leave the host. Rather than the females, the first instar larvae are the mobile stage that seek out new hosts. Parasitoids can be further categorized depending on where they occur on the host. For example, parasitoids that develop within the host are referred to as endoparasitoids, while those that live on the host while feeding are called ectoparasitoids. The impact of the parasitoids on the host can also vary by species. Parasitoids that develop while the host continues to feed and grow are called koinobiont parasitoids. Other parasitoids, known as idiobionts, paralyze and inhibit further development of their hosts while accessing the host tissue from the outside. Idiobionts are almost exclusively ectoparasitoids. Parasitism itself can occur in different ways and even at different trophic levels, as not all parasitoids attack herbivores. Superparasitism occurs when a host is attacked multiple times by individual parasitoids of a single species. This usually happens when there is a high density of parasitoids relative to the hosts, or when the parasitoid has small eggs and high fecundity. For the most part, parasitoids avoid superparasitism as it increases intraspecific competition and reduces fitness. Most parasitoids use chemoreceptors located on their antennae and ovipositor to identify and avoid hosts which are already parasitized. Multiparasitism, on the other hand, occurs when a host is parasitized by multiple species of parasitoids. Multiparasitism is more common than superparasitism. The interspecific competition among parasitoids evoked in multiparasitism usually results in the successful development of only one species of parasitoid. Sometimes, the larvae or pupae of a parasitoid serve as the host for another parasitoid species. This interaction at the fourth trophic level is called hyperparasitism. Hyperparasitoid larvae feed on other parasites or parasitoids associated with the original host. These insects may interfere with pest management programs in which parasitoids are used as biological control agents. Endoparasitic larvae live and develop within the arthropod host. Their survival is dependent on the parasitoid's ability to evade or suppress the host's immune responses. Meanwhile, a host can use different methods to defend itself against parasitism. A common defense mechanism used by insect hosts is an immune response known as encapsulation. During encapsulation, host hemocytes act in concert to surround the parasitoid, forming a capsule that kills the parasitoid egg or larva by cutting off its access to oxygen and nutrition. But, as with any interspecific interaction, parasitoids can evolve to overcome host defenses. In some dipteran parasitoid species, the host's immune system is hijacked. Instead of killing the larva, the capsule becomes a protective sheath. Some parasitoids even maintain mutualistic relationships with viruses that can help suppress host immune responses, which promotes successful parasitism. Polydnaviruses, for example, have evolved specific symbiotic relationships with parasitic wasps in the families Ichneumonidae and Burkonidae. The viruses reside and replicate within the ovaries of the female wasps and are injected into the body cavity of their caterpillar hosts along with the wasp eggs. Genes expressed by the viruses help to suppress host immune responses, which allows the larval wasps to successfully complete development inside the host. The viruses are also known to alter the host metabolism and development to create conditions that are beneficial for growth and survival of the parasitoid larvae. Other endoparasitoids evade the host immune system by carefully placing offspring in certain positions within the host's body. They usually target specific tissues, such as the fat body or the ganglia of the central nervous system. In these locations, eggs are shielded from host immune responses. Parasitoids have co-evolved with their hosts and are not only able to manipulate the host's immune system, they often hijack the host's nervous system as well. In some species of parasitoid wasps, the larvae manipulate the host's behavior for their own benefit. Some ectoparasitoids of spiders induce particular behaviors in which the parasitoid wasp causes the spider host to spin a special type of web that the wasp larva then turns into a cocoon when it is ready to pupate. Other endoparasitoids induce a behavior in the host known as bodyguard manipulation. This occurs in some braconid wasp species that pupate outside of their caterpillar host on external substrates. The host is kept alive even after the larvae emerge to pupate. If disturbed, the caterpillar violently thrashes from side to side to defend the wasp pupae against predators and hyperparasitoids. Some wasp species even manipulate the caterpillar host to seek out shelter or to spin protective cocoons over the parasitoid pupae. The next biological control agents that we will discuss are the predators. Predators are free-living organisms that feed on other animals and usually rapidly kill their prey. Most predators require more than one prey item to complete development and can attack and kill several animals during their lifespan. Unlike parasitoids, most insect predators are generalists and can feed on multiple prey species. Common examples of arthropod predators used as biological control agents include ladybird beetles, predatory mites, and lacewings. Ladybird beetles are used to manage aphids and herbivorous mites. Lacewings are voracious and effective predators of aphids. Predatory mites are used to control herbivorous mite species. These three common biological control agents are predators in both the larval and adult stages. Predators are most effective as biological control agents when pest population densities are low. Because predators are generalists, a diversity of predators in a managed system will provide the best, most balanced control and permit host switching by generalist predators without the loss of control. Although not commonly used in biological control, you're likely familiar with another group of predatory insects, vespid wasps. Far from just a nuisance at picnics, these insects have an important ecological role as most vespid wasps, such as yellow jackets, are voracious predators. These wasps contribute to the control of many herbivorous insects, including some pestiferous caterpillars. So far in this lesson, we have focused on the use of parasitic and predatory biological control agents of arthropod pests. Pathogenic microbes such as bacteria, fungi, and viruses are also natural enemies that can infect arthropod pests. Formulations of some of these pathogenic microbes are commercially available as biological control agents against insect pests. Bacterial formulations are among the most common microbial insecticides used in biological control. An example of a widely used bacterial biological control agent is Bacillus thuringiensis, or BT. This soil-borne bacterium has insecticidal properties that have been harnessed in a number of different formulations. Different strains of BT are toxic to different types of insect pests and therefore can provide targeted control. As you may recall from Module 9, this bacterium produces crystals which contain endotoxins that, once ingested, damage and paralyze the midgut of the insect. This causes death by starvation and infection. BT can be applied in a variety of ways. It can be administered on the surface of plants from emulsified concentrates or wettable powders, and it can be applied in aquatic systems as a granular formulation to control mosquito larvae. The gene that produces the toxin can also be genetically introduced directly into crop plants so that the insecticidal toxin is expressed throughout the plant tissue. In addition to bacteria, fungi and viruses can also be used as biological control agents. Over 750 fungal species naturally infect insects and thus contribute to the regulation of insect populations. Unlike most other microbes, fungi can attack insects through intact cuticle. Fungal spores attach to the insect cuticle, germinate, and penetrate the integument, gradually infecting the hemocoel and often killing the insect in the process. The fungi usually produce toxins that incapacitate the insect, and the toxins may even kill the insect before the fungal growth does. A good example of a commonly used fungal biological control agent is Beauveria bassiana. Spores of this fungus can be applied to control many different insect pests, including whitefly and aphid pests in greenhouse operations. Formulations of bacterial and fungal biological control agents that act as microbial insecticides are becoming increasingly important, as they can be applied for rapid responses to outbreaks of pest populations while reducing the introduction of harmful chemicals into the environment. These control agents have very little impact on non-target insects and can be mass-produced for wide-scale applications. There are also many different types of viruses that can be used as biological control agents. Baculoviruses are pathogens that attack insects and other arthropods and include a wide range of viruses that attack specific insects. These viruses must be consumed by the insect, and once ingested, virus replication within the host cells ultimately kills the insect. From the outside, the insect can appear to dissolve. As it falls apart, it releases the virus onto the surrounding surfaces, where other insects can come into contact with it. Baculoviruses have been formulated to target specific insect pests. For example, a formulation was developed for the major forest defoliator, the gypsy moth, an invasive pest of hardwood trees in North America. This formulation was registered in 1978 in the USA and has been sprayed over thousands of acres of forest to control the gypsy moth. Another baculovirus formulation has been developed to target the important cosmopolitan pest of tree fruits, the codling moth. This virus formulation is applied widely to around 150,000 hectares per year in tree fruit orchards in the USA. These virus formulations are species-specific and break down in the environment quickly, allowing them to fit in well within IPM programs. However, this species specificity is also part of the downside to baculoviruses for pest control. Development costs are high, since each formulation needs to be developed for each pest species. Additionally, most viruses can only be developed inside living insects, which is an expensive way to produce a large amount of formulation. Growers also want to minimize costs and prefer insecticides that can manage more than one pest species at a time. These challenges have precluded extensive use of virus formulations in commercial pest management programs. We have discussed how the introduction of bacterial, fungal, and viral pathogens can be used in pest management against insect pests. However, have you considered the possibility of attacking the insect by targeting the symbiotic microorganisms it already carries? Recall that most, if not all insects, house microbial symbionts within their bodies. Some of these symbionts can be beneficial, while others are benign. Together, these microbes form what we refer to as a microbiome, a community of microorganisms that protect the host against diseases, produce essential vitamins, and assist in digestion. By disturbing or manipulating the microbiome of an insect pest, we can inflict different kinds of effects on the insect, such as decreased growth rates, diminished reproductive success, or even a reduction in the ability of an insect to transmit diseases. There are 3 techniques currently under development for this pest management tactic. The first involves the introduction of novel microorganisms into insect microbiomes. For example, the introduction of bacteria in the genus Wolbachia into Aedes aegypti mosquitoes affects both the mosquito's reproductive success and its ability to transmit dengue fever. The second method is to genetically modify microorganisms already present within the pest species. The microorganisms can be modified to produce insecticidal compounds that can kill the host insect. In one study, genetically modified bacteria were successfully introduced into the vectors of Chagas disease, a tropical disease spread by insects commonly known as kissing bugs. the modified bacteria interfered with natural gene expression within the disease vector. This caused a disruption of protein synthesis, a substantial reduction in oviposition success, and increased juvenile mortality of kissing bugs. The third and final technique is to target and eliminate essential microorganisms in a pest's microbiome. By removing populations of symbionts that the insect pest relies on for growth, reproduction, and survival, we can reduce insect pest populations and thus reduce the amount of damage they inflict. Currently, the manipulation of endosymbionts in biological pest control remains a relatively novel pest management strategy. While it has shown promise both in laboratories and in field experiments, further research is needed to improve its effectiveness, specificity, and sustainability. Biological control agents can also be applied in weed management. In this case, the natural enemies of the pest are not parasitoids or predators, but herbivorous arthropods. Some plant pathogens can also act as biological control agents of weeds. Weeds are unwanted plants that grow in competition with other plants and crops. They can also be poisonous to animals, including humans. In extreme cases, large populations of a single fast-growing weed species can lead to the local extinction of native flora and upset the ecological balance of a region. Fortunately, herbivorous arthropods can be used as biological control agents against weeds. As these insects feed on different parts of weedy plants, such as the flowers, roots, or seeds, they can help keep weed populations under control. Biological control of weeds involves a lot of research, as the natural enemies need to specifically attack only the weed species. A good example of weed control using herbivorous insects is the program for control of yellow and Dalmatian toadflax using introduced weevils in Alberta, Canada. Let's talk to Dr. Rose de Klerk-Float from Agriculture and Agri-Food Canada, who will tell us more about the research behind the successful program.
I'm a research scientist, and I'm an entomologist in biological control of weeds. So our program goes back to actually 1951. And since that time to present, or I guess last year, we've released 81 different insect species, I should say arthropods, because there have been some mites as well, against approximately 30 different weed species. Because weed biocontrol typically doesn't work that quickly. It typically doesn't outright kill individual plants. What it does typically is reduce the vigor of a plant and the reproduction potential, and in that way limits its impact. Like what we're trying to do, we're not trying to eradicate, we're trying to bring the populations down to below a certain threshold, whether they're economic thresholds or environmental. And I want to emphasize that it's just one tool in, among other tools to mitigate these weed problems. And ideally, we should be looking at more ways to integrate biocontrol with other methods, such as chemical control. And so one I've been involved with is Dalmatian toadflax, particularly in British Columbia on rangelands. And we've released a stem-boring weevil that has been very effective at controlling that weed. What makes a plant or any organism invasive in a new environment is that they have arrived here either intentionally or accidentally, often accidentally, without the natural enemies that keep them in check in their place of origin. The thing is that they also have the capacity to reproductively increase quite rapidly. They have a high rate of reproduction. And, like a plant, too, that's highly competitive can grab resources away, whether that's light resources or nutrients in the soil. They often have that capacity to take away those important elements for other plant growth for their own, for their own growth. So that's why we go to biological control, and we also go to biological control when the weed, after the weed has just taken off and has spread so widely that you have to do something, but it's not feasible to use chemicals. It's not feasible to use other methods like cultural control, where you're tilling or cutting or pulling. Like, you just couldn't do that. There's yellow toadflax, Linaria vulgaris, and there's Dalmatian toadflax, Linaria dalmatica. But yellow toadflax has always been a more difficult target. It grows very differently from Dalmatian. It tends to creep versus Dalmatian that grows more erect and doesn't have that creeping feature. And whereas Dalmatian's more succulent and adapted to drier habitats, which means it does quite well in the dry rangelands of British Columbia. Yellow toadflax is a weed at both rangelands, but also croplands. Actually, yellow toadflax, it occurs throughout Europe and into Asia as well. So then we went on to a stem-boring weevil. And this is the story where we thought we had one species of stem-boring weevil. At that point, we called it Mucinus ianthinus. And we thought it could attack both Dalmatian and yellow toadflax. And we started releasing it, thinking, we've got one agent that can attack these two really bad imported weeds or introduced weeds. And so it's only been recently that we realized that we had two species. So yeah, those are the plans. Oh yeah, and now we have this brand new agent that was released first in 2014. It's a gall-forming weevil, forms a big gall on the stems of toadflax, yellow toadflax. And I think it has a lot of potential. Like, it's the early stages, but it has a lot of potential. With every new imported insect, there is a risk that this insect may feed on other desirable vegetation, especially if it's closely related to the weed. So that's why we try to be responsible in bringing in as few and as effective agents as possible. That the risk is very, very low. And a lot of it has to do with how gall-forming insects in general have evolved such an intimate relationship with their host plants. But the one thing that really clenched the safety of this insect was a cross-section showing the egg being literally displaced and spit out of a plant that wasn't its host. Because the whole idea is that we don't want to have to repeatedly release this insect. So if you have too few that are emerging initially, and then they start dispersing wide and far, they can't find each other to mate. So that's another issue. So you also want them to stay put for a while so they can have an impact on the local weed patch before they start finding a new patch of weeds. So I would say that it's showing great promise. It seems to be building in number now at some of our older sites. But we're only talking about 4 summers, So I think the next stage we'll be looking at whether or not it can have an impact on its host weed. So that's where we're at with it. It's Mogilones crucifer. It has a little cross on its back, and it's been such a star, And yeah, that's my favorite at this point, anyway.
Now that you have a better idea of the different types of biological control agents that can be used in pest management, let's learn about how natural enemies can be implemented for biocontrol. In the next video, we will explore 3 different approaches to biological control.